Afternoon, guys. So this afternoon we are going to start our animal physiology lectures finally, right? So this is going to be the first lecture. It's on chapter 1. Chapter 1 is theme-based, basically, talking about kind of animal physiology, what a mechanism is, what feedback systems are, and whatnot. That's kind of what we're going to go through today. So this is what's going to be covered today here. What is animal phys? That's what AP stands for. What mechanisms are, what the roots of animal phys are, their expense, what structure and function is, what homeostasis is defined as, tie homeostasis into feedback loops, and then we'll do conformers versus regulators. So got a beautiful octopus up here, which is known as a cephalopod, part of gastropoda. That group is a strict regulator for an invertebrate. That's pretty rare, and we'll kind of talk about what that means when we get to that part of lecture. So physiology. I love physiology. I'm a physiologist and an anatomist. So I'm a trained comparative anatomist. I taught physiology in graduate school, done some physiology research, which is really hard to do. But physiology is awesome. It is really, really neat, particularly when you start talking about non-mammalian vertebrates, that they have all these different, very interesting physiologic adaptations, if you will, to environments, particularly extreme ones. So animals do incredible things. I'm a herpetologist. I study reptiles and amphibians mostly. And this particular guy down here, this turtle, is a painted turtle. These guys, they're active under frozen ponds, and they're moving around in the spring. They even reproduce in the spring in northern Michigan, and Michigan in particular, under the ice. So that's pretty amazing. How do they do that in that low oxygen situation and things are so cold? Turtles are also found from the subtropics all the way through the tropics up into Antarctica. So they are, or I should say the subarctic, not Antarctica, subarctic, which is pretty incredible. They are a fantastic species. Some of the frogs here in Indiana completely freeze. So the gray tree frog, which is one that's found up in Indianapolis and down here in southern Indiana, they dig just under the leaf litter and completely freeze solid like an ice cube. Their hearts stop, and then they wake back up in the spring and continue their life. How do they do that, right? The physiology behind that is really amazing. And so physiology, if you look around, is quite interesting. So what is animal physiology? How's it different than, say, a mammalian physiology? So animal physiology, we're going to take a comparative evolutionary approach. We're going to look at lots of different organisms, both vertebrates and invertebrates, and how they deal with life, right? How they adapt with life, and what physiology behind or within their bodies is utilized by evolution to make sure that they survive and reproduce. So we're going to focus on the functions of tissues, organs, and organ systems within multicellular animals. A lot of what we're going to do is vertebrate physiology, but we'll use some invertebrate examples as we go along. Physiology is an integrated science. It happens at all levels of organization. And I know you've seen a slide of levels of organization where we start with the chemical level, the actual elements and atoms combine to form molecules and compounds. Those molecules and compounds are going to combine into membranes and membrane-bound structures, which form organelles and cell membranes of cells. Cells are organized into tissues, tissues into organs, organ systems into the organism. You can study physiology at any of these levels. Also, what makes physiology quite challenging, if you will, and sometimes difficult, is there's a lot of chemistry and physics that's involved with understanding physiology. Not that I'm going to make this a chemistry and physics class, but we're going to utilize the principles of the physical world because we have to abide by the principles and those laws that control the physical world. We got to manipulate them, modify them, and in some cases, just leap above them in order to survive. So we'll deal with looking at particular aspects of physics and chemistry to understand physiologic mechanisms. So the types of questions that you see in animal physiology are what mechanism is functioning to accomplish what the organism's doing. So when we go back to those frogs, how do they freeze without dying, without damaging their cells? How do they wake up? Some of these questions have been answered, some of them have not. I don't know, people are still studying them. And then we want to know, how does that mechanism come to be? That is, what's the evolution behind it? Are there some clues out there in other organisms that allow us maybe to understand the evolution of the mechanism in interest? Now, in what is a mechanism, right? So I keep saying this word mechanism. It's a component of all living organisms and the interaction among components within the body that lead to a particular function or enable an animal to perform a function within the body. And mechanisms are central to physiology. And we'll talk about lots of different mechanisms. Matter of fact, I'm going to define what homeostasis here is in a few minutes and then give you some mechanisms by how we control it. I'm also going to tell you about lightning bugs, right? And I love lightning bugs. If you're from the Midwest, I caught them as a little kid. And, you know, have you ever thought about or asked the question, and I did when I was little, what makes a lightning bug light up? And then why does it do it, right? Those were questions I asked my parents who had no idea why or how. Okay, so let's talk about a mechanism so you can see how a mechanism works. So, as I mentioned, I love fireflies, and I always have. And I wondered as a kid, how do fireflies produce light? And why do they produce light, right? So the how and why, if you will. And this is actually the mechanism that's in your textbook. And it's pretty neat that they figured this out. There are a couple different compounds that are involved, right? The first one's called luciferin, as you can see up here. So firefly luciferin is produced in the light organ. This is what the light organ looks like on the inside of the insect. The light organ, you know, is located on the caudal end or the tail end of a firefly. And if we look inside of that, this is what it looks like. So there are a bunch of rows of cells here that are lit up, and there are some conducting tubes that are going to branch and come really close to each of the individual light cells. These tubules are gas transporting tubules, and they're called tracheids, are the name that they give them. So tracheids are the name of these tubules that deliver oxygen to the cells in insects. Insects have an open circulatory system, and therefore they don't have the closed blood vessels like we do. So whereas our cells are only a couple cells away from a blood vessel, a capillary is what we call it, the smallest blood vessels. In insects, there are really small tubes that look like very minute tracheas, if you will, the windpipe, that deliver oxygen to cells. And we're going to talk about why oxygen is important here in a minute. But these cells that line these long tubes within the light organ produce luciferin, which is a protein. And this luciferin, what it can do is it can bind to ATP and split it. When it splits ATP, it is going to bind, it's going to release a couple phosphates, and it's going to bind AMP, adenosine monophosphate, there's only one. ATP has three, right? So it releases two of the phosphates, keeps one of them around. And what we've done is stored some energy in that chemical bond there, that covalent bond. Now, when oxygen's available, there's an enzyme called luciferase. As you can see here, I've underlined the ACE. All right, whenever you see ACE at the end of a word, it means enzyme. Luciferase, what it can do is it can bind onto luciferin AMP, add the oxygen, and then it can split off the AMP, and that excites the luciferin. It excites some of the electrons in the luciferin, which when you excite electrons, they jump up to an orbital they don't belong in, and then they kind of come back down. And as they come back down, they have to release the energy that excited them. In this particular organ, and with the way luciferase works, it releases photons or light energy, and that light escapes as a wavelength that is usually yellowish to green, right? And that's how they produce light. So when oxygen's available to the light cells, and it floods the light cells, and is not used up by the mitochondria, then it will run this reaction with luciferase, oxygen and luciferase, and luciferin and AMP. Now, the question is, fireflies flash their bulb, right? They flash their light organ. They flash it at certain speeds, at certain cadences. And what the reason they do that, you'll learn in a minute. But how do they do that? How do they control the oxygen, right? We know these tubes deliver oxygen, and you guys all remember why cells need oxygen, right? Cells need oxygen because the mitochondria utilizes it to make ATP. And if you remember from 165, the end of the electron transport chain, the final acceptor of the protons that are passed along all of those proteins of the electron transport chain is oxygen, right? That's why we breathe in oxygen. We breathe in oxygen, it circulates through our blood with red blood cells. We drop it off the cells. The oxygen enters the mitochondria, binds to the end of the electron transport chain, and draws those protons to it, right? So we can make ATP. And so typically in a normal cell, if oxygen comes from the tube, it enters the cell and is going to enter the mitochondria and help run ATP production, right? The problem with the light cell is if all the oxygen that comes in is used up by the mitochondria, there's nothing to run this reaction that produces light because we need oxygen, right? So what these cells can produce is nitric oxide, all right? And there's an enzyme called nitric oxide synthase, all right? And what it does is it takes this amino acid and produces nitric oxide, all right? And nitric oxide is NO, all right? And NO is used as a localized kind of cell ligand, a localized cell communicator. It tells, in vertebrates at least, it tells, for example, smooth muscle to relax. And cells around the smooth muscle will relax. Cells around the smooth muscle in blood vessels, for example, will produce nitric oxide, causing the smooth muscle and blood vessels to relax to deliver more blood, for example. And that's kind of how nitric oxide works in vertebrates. Nitric oxide is a gas. So you can activate this enzyme, nitric oxide synthase, in the light cell. It'll produce nitric oxide by using some of the amino acids that are pretty common in every cell. The nitric oxide, what it does is it enters the mitochondria and binds where the oxygen would, and then prevents oxygen from binding. And it shuts the electron transport chain down for a few seconds, for example. What that does is prevent the oxygen from being used by the mitochondria. Then the oxygen can catalyze, if you will, this luciferin AMP to luciferin light release, right? So when the lightning bug wants to flash, it produces a nitric oxide, a burst of nitric oxide. That allows oxygen to reach the luciferase and you get a flash of light. And then it'll remove the nitric oxide. Nitric oxide is a gas. It can flow in and flow out. So it stops the production of it. And then you go dark again. And then you produce a burst of nitric oxide and then light again. So every time you get a flash within the lightning bug organ, it's a burst of nitric oxide that causes it. And that's kind of the mechanism behind this. They can also control how much oxygen is entering. That can help out also. But these are kind of the mechanisms behind light production and light flashing in lightning bugs. Really, really cool, right? Really neat. So the big question that we haven't answered is, okay, that's the how, that's the mechanism, right? But why do fireflies do this? And I've always wondered that also. And we'll get back to that here in a few minutes. All right, so drop my eraser. So, a couple of other things before we come back to lightning bugs. Physiology is rooted in chemistry and physics. So I told you it's an integrated science, and that's kind of what makes physio hard, right? Is this linkage to the physical world. And we have to abide by the laws of the universe. If we want to circulate blood, if we want to move air, we got to change volume. We got to change pressure. We got to deal with pressure. If we want to circulate blood, we got to produce pressure, right? If we want to move water, we have to abide by the laws of osmolarity. So there are a whole bunch of principles that are based on physical chemistry, or organic chemistry, general chemistry, and physics. And we'll talk about a few of these. Now, I promise to make it interesting, right? I'll be honest with you. When I was an undergrad, physics and chemistry were very boring. I did not like them. I had to struggle through them. I had to make myself study them. It wasn't until I got to grad school and started studying physiology and incorporating the principles of chemistry and physics into a biological system that chemistry and physics became really neat, really interesting. And hopefully that'll happen here with you guys. So if you look at this table from an old physiology text, nerve conduction, ionic current, Ohm's law is important, a law you learn in physics. Boyle's gas laws are important in gas exchange. Those are laws that you learn in chemistry class. Gravity, inertia, momentum, velocity, drag, vectors, the law of thermodynamics, all of those are utilized by the body. We have to build or adapt our bodies to meet those physical principles that the universe places on us. So physiology can be understood in terms of physical constraint. And so, for example, you might think, why aren't we just one big cell, right? Why not just be one big cell instead of millions of cells? And it becomes really, really readily apparent as the cell gets bigger, its surface area to volume decreases. That is, the surface area you have is a lot less than the volume of the cell as it gets bigger and bigger. That's why our cells aren't very big. We might as well take a big volume and divide it into smaller volumes by becoming multicellular. That's why multicellularity is so common among large organisms, because we need to divide up the cytoplasm enough so that we have more surface area to volume, which will allow us to move things more efficiently at our surface so that our cytoplasm has the proper amount of nutrients, oxygen, and whatnot that it needs to run everyday life. So physiology, physics, and chemistry are indivisible. when it comes down to it, you need to manipulate chemistry and physics in order to survive. And so we will apply some of these principles of chemistry and physics to our mechanisms. But I promise to keep it cool, or keep it interesting. Evolution in animal physiology. Obviously, animal phys is embedded in evolution like every biological field, right? So central concepts of evolution, speciation, natural selection, Darwinian evolution, they're all the underlying principles of why physiologic mechanisms have evolved. And there are a bunch of really cool examples of natural selection happening at the physiologic level of biology. And I just list a few here that are really, really neat. So mammals and birds, I don't know if you think about this, we have really high body temperatures. You maintain a body temperature of 98.6 degrees. A lot of invertebrates don't do that, or a lot of vertebrates don't do that, right? Because we maintain such high body temperatures, our enzymes had to evolve to tolerate high body temperature. I mean, that's really important. You know, the proteins found in a frog cannot tolerate, the enzymes found in frog blood, for example, or in frog interstitial fluid, cannot tolerate the high body temperatures that mammals have. That's because our enzymes have evolved over the millions of years of evolution in birds and mammals to tolerate high temperature. There are crabs, land crabs. You typically think of crabs that live in water, right? Well, think about hermit crabs. They still have gills like other crabs, but their gills have evolved to work in air. Migrating salmon and several other types of fish can move in and out of fresh and salt water because their bodies tolerate the differences between fresh and salt water as far as osmolarity goes. There's even one species of frog that lives in salt water. Probably didn't know that. It's the only one. And so the big question is why? Why would a frog want to live in salt water? The answer is pretty simple. It's driven by evolution, right? What is the whole principle of evolution is to adapt to survive, get enough energy to reproduce. And if I can reproduce and pass it on to offspring, then I have good fitness, right? It's the concepts you've hopefully learned and heard in 155, 165, or if you've taken evolution with Dr. Milne, for example. So this frog, which can live in salt water, is the only frog can do that. It has a niche that no other animal in its group, its taxa, right, in amphibia, class amphibia, can accomplish. So it has unlimited food sources for amphibians. And so it survives really well because its tadpoles, eggs, and the frog itself, which is called the crab-eating frog and lives in Indonesia, can actually survive on islands, for example, where there are no other amphibians. It's really neat, right? Really neat stuff. So physiology and how these organisms do that are based on evolution and natural selection and adaptation. If we go back to the lightning bug, you can see this, right? So we talked about the mechanism of flashing in light, and that's really neat and cool, but why? Why do they do it, right? And so this is a pattern of flashing that a biologist had studied on different species of fireflies. So fireflies, fireflies produce light. What is the adaptive significance of that? Why do they do it? How does that make them adapt to their environment? Well, we know these guys come out at night. They also feed at night. The physiologists figured out the mechanism. The evolutionary biologists figured out the reason why. So they figured out pretty quickly that different species of fireflies have different brightnesses to their light and different speeds and cadences to how they flash. And that type of flashing is what attracts their mates, or says, hey, Jimmy, I'm over here, right? And a species can, a male and female, or a population of lightning bugs can find each other while there are several other species of lightning bugs out in the forest, can find each other by flashing, right, at a certain speed and cadence. This allows mates to find each other. Fireflies also mimic other fireflies and their cadence so that they can predate on them. So you can imagine if I'm a firefly over here and there's a smaller species flying around and I mimic their cadence and their flashing, that little guy comes over, I have a food source, right? I have dinner, if you will, for the night. And so they utilize it in that way. It attracts mates, but it also attracts prey, right? And so that's why lightning bugs do this. So that's the adaptive significance of the mechanism and the reason why it showed up in the first place, right? Right? It evolved in these particular insects. All right, so the other thing that makes physiology really, really interesting is it's really, really expensive. So as a biologist and a physiologist, sometimes I wonder, why does an organism do that when you know it's really, really expensive? If you look inside of me or you or another animal out there, another vertebrate, you'll find a bunch of organs, right? And have you ever asked the question, why aren't we just like one big gonad, right? Because if all we have to do is reproduce, why not just be one huge ovary or one huge test that's produced tons of gametes and then, produce offspring? Why don't we just do that? There are some animals that do. There are some invertebrates that do it that way. And the reason we don't do that is because all of the organs that you see in the body allow us to adapt to certain niches and environments, which then allow us to manipulate a food source that other animals can't, which then allows us to reproduce, right? So we build this capital, if you will. We invest a tremendous amount of development and energy into maintaining these really amazing organs, right? And each of these organ systems is not only coordinated with the others, but is responsible for doing something that helps our internal environment, the inside of us, maintain its constant conditions no matter what chaos is happening around us, right? And that's really why mammals and many vertebrates are what are called regulators. All of the investment in the organs and then all of the expense of the overhead of running those, all the energy that is required to running those, and particularly our high body temperature as mammals, allows us to maintain very specific conditions in the body where everybody's happy, all the cells and organs are happy, and then we invest a little bit of the energy left over into our reproductive organs so that we can produce gametes and offspring. And if you can take advantage of an environment that other animals can't because of the organs you have developed and the mechanisms of physiology that occur within them, then you can survive to adulthood and pass your genes on. And that is really why physiology is expensive. And those are the two questions at the bottom. So why is physiology expensive? Because of capital and overhead. And then is it worth it, right? And it is worth it if the organism can reproduce and produce offspring, right? So animals are significantly different internally than they are to their environment. We're going to call it homeostasis. All right, our homeostatic conditions within our body are a lot different than the air around you and the temperature around you. The capital that we build to maintain these very different environments, our inside of our body to the outside environment, is called capital. Things like kidneys and gills and livers and lungs and trachea, all of the stuff that we built in our body outside of the gonads that are responsible for maintaining our everyday activities. It's called capital. The overhead is the metabolism that is involved in maintaining those organs and transporting ions and running physiology. Allows us to survive in our environment, allows our insides to be very happy and under consistent conditions if we're a regulator, no matter what's going on outside. So for example, this fish here is gaining salt, it's losing water, it's trying to conserve water as it excretes it. Salt water doesn't drink salt water, so how does it maintain its proper water concentration? That all is entailed in its physiologic build and its organ makeup and how those organs perform within the fish that allows it to survive in the aquatic environment, particularly the aquatic environment we call the ocean, right? Some fish can move back and forth between the ocean and fresh water, which is an amazing feat in order to reproduce. So that's kind of why physiology is expensive, which helps us understand why physiology is important. Because it's expensive and because we want to understand how organisms can survive in these really extreme environments on an everyday basis, we have to kind of understand how selection works and then how these mechanisms work and what the adaptive significance of the mechanisms are, like we talked about with the lightning bug. So efficient physiologic systems allow us to survive. They allow us to preserve a little bit of energy for reproduction, and then we can pass our genes on to the next generation. This is called survival of the fittest. A fit organism has the ability to produce offspring that can then produce offspring, that can then produce offspring, and continue that line of evolution for generations to come. All right, so as you can see here, some of the central themes here that we've talked about and have mentioned here, I just wanted to put on slides for you. So the major goals here is in this course for this semester is to explore kind of what mechanisms are found between taxa. Taxa means group. So you can see up here with my pointer, taxa means group of animals. Show how selection has kind of shaped the phenotype and therefore the function of certain parts or even the entire organism as we talk about physiology of maybe occupying different environments or of organ systems. What's really cool about comparative physiology, or what we call animal physiology, is you might find several animals in a particular environment, and all of them have solved a problem of living or existing in that environment in different ways, which is kind of neat to do. And the big question that a lot of people have asked me in the past is, are one of those mechanisms better than the other? And the answer is really no. If an animal is surviving within an environment and producing offspring, then in reality, it's doing quite well. Its fitness is quite high. It doesn't matter how it does it, because different lines of animals, or different evolutionary lines of animals, will have different mechanisms for which they actually survive, or have different adaptations, phenotypes that lead to different mechanisms that allow them to survive in niches and environmental conditions. And as long as they can survive and reproduce, then evolution is doing its job. We'll also compare the different ways animals adapt as we go along. And we will also study form and function, all right? So number one here, form and function, or mechanisms. What something looks like often determines what the mechanism is going to be. So I will spend a lot of time with you in the beginning of each of the new topics that we're going to cover, studying the structure of whatever organ or tissue we're talking about. And that's why histology becomes really important. So that's why we're doing a histological review for that first lab, so that you guys have a good foundation, a good review of histology, so that you all will do well when I start going over organs that have epithelia, nerves, and muscle, and what type of muscle it is and whatnot. And then we can study the mechanisms that structure, that architecture will help determine. We're also going to come back many times talking about homeostasis, because really a lot of the capital that we invest in the organs, or the capital we call organs, and the metabolism that we use to run it, is really helping us as regulators, particularly most vertebrates are regulators, to maintain homeostasis, right? And homeostasis is the, as we're going to see a slide or two here, is the basic conditions within the body that are, remain constant at all times, so that our cells are happy and perform very well. Homeostasis is maintained by feedback mechanisms or control systems. We're going to talk about those. And then we'll talk briefly about conformity and regulators here to finish the lecture off. So form and function. And again, as I mentioned, I'll spend a lot of time doing some anatomy, some microscopic anatomy, because structure often depicts or tells you what the function is going to be. So functions are influenced by structures. And you can use this example here of frogs and muscle types. So you got a nice little frog here, a red-eyed tree frog hopping for a fly, as you can see up here with the laser pointer. And there are 3 different muscle types that are going to be involved with this. You got skeletal, cardiac, and smooth muscle. And if you know anything about those muscle types, they kind of look different from one another, and they perform, therefore, slightly different functions. So skeletal muscle that's stretched across the legs here of my frog allow him to move in the environment, right? The elongated pelvis and the elongated femur stretches the muscle so that they can spring and fire from a sitting position really nicely and catch, for example, their prey, flies. The skeletal muscle in the tongue allows them to flip their tongue out and grab onto that fly, right? Once they grab onto the fly and swallow it, the fly is going to end up in the digestive tract, which has a different muscle type, smooth muscle, which contracts completely differently and in a different sequence. It's more of a contracting wave-like motion that smooth muscle produces, which is going to macerate and mix and then push the food through the digestive tract. Once the food is completely digested, it enters circulation, and what is forcing blood through the circulatory system in a vertebrate like frogs is the heart. And the heart's made up of cardiac muscle, which again has its own properties and therefore its own function compared to smooth and skeletal muscle. So even though there are some similarities in the muscle types, they both have actin and myosin. These are your motor proteins. They are kind of organized, and the architecture is slightly different in those 3 muscle types, as we'll talk about when we get to muscle tissue, that allows those types of muscles and their location in the body to perform the functions that they are responsible for. Okay, let's talk about homeostasis. So homeostasis, the word literally means same stasis. Homeo means same, stasis means static conditions, right? Animals maintain very constant conditions inside of their body. The inside of the body is going to be called the internal environment, all right? There are a couple different body fluids that we see in animals. We have blood, right? Or sometimes it's called plasma. Then we have cytoplasm, right? And we have intercellular fluid. It's also called interstitial fluid, right? The interstitial fluid is called the internal environment because that's the environment the cells are bathed in. They're actually sitting in that fluid. So homeostasis happens here, happens in the interstitial fluid. That fluid maintains very consistent conditions for the cells, and those conditions have evolved with those cells as the cells have evolved through time, and they really like their conditions in a certain way, in a regulator. So therefore, all of our cells in our body are in a temperature of 98.6 degrees. All the cells in our body have a certain amount of glucose that surrounds them, a certain amount of oxygen, a certain pH, a certain salinity. Those things are maintained no matter what's happening on the outside of our body, whether the temperature changes, it's raining, it's snowing, you drink a lot of salt water, you drink fresh water, you drink a Coke, a Gatorade, all of these things that are coming into your body and surrounding your body, which can be very variable and chaotic, do not affect the internal environment of your body, which is maintained at very consistent conditions. That's called homeostasis. And all of the organ systems play a part in helping maintain those conditions. So the intracellular environment, intra, as you can see, is the cytoplasm. So intra is cytoplasm. Intra, this word intra right here, means inside of the cell. Extracellular fluid is on the outside of the cell. Blood and interstitial fluid are extracellular fluids. There's another fluid that's an extracellular fluid that we call lymph. And the name lymph, interstitial, and blood all come from where they're located. Location, 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 right? Now, there was a French fella here, a French physiologist, Claude Bernard, who actually coined the term constant internal environment in French. Later on, it was called by Walter Cannon and some of his colleagues homeostasis, which is universal along living systems. Walter Cannon is really responsible for applying this internal environment or these constant conditions not only to cells, which is what Claude said. He said that, the internal environment, the intracellular fluid, the cytoplasm, is held in a consistent state. Cannon applied that to tissues and to the interstitial fluid in all organs and body systems. And so homeostasis is also universal in living organisms, particularly those that are regulators, that are like vertebrates, for example. And no matter what's going on the outside, you can see this large external fluctuation in temperature, salinity, water content. Nothing is really happening, or very little change happens inside of the regulator. And that's because your organ and organ systems help maintain those conditions. The way they do it is through feedback mechanisms. Your body, particularly the nervous and endocrine systems, which we'll talk about later, are going to monitor something called controlled conditions, which you can see right here, right? So what are the controlled conditions? These are basically conditions which in the body that are held constant. And your body monitors those conditions, organs, tissues, particular receptors, particular parts of the nervous system. Their job all day is to monitor these control conditions. So they're monitoring things like oxygen concentration. They're monitoring things like carbon dioxide concentration, sugar concentration, salinity, water. There are thousands of these control conditions that the nervous system and these receptors and sometimes specialized organs or tissues are monitoring. And if there's a change in the control condition, the concentration of glucose drops or increases, our body temperature goes up or down, it is going to be stimulated, right? It's actually going to respond to that change. That's what we call a stress. And it is going to bring about events within the body that reverse the stress, right? So if you get an increase in body temperature, you're going to sweat, right? Which decreases body temperature. If you decrease body temperature, you're going to shiver to increase body temperature. And so there are several organs involved with the nervous system that help maintain these control conditions. And that's kind of what we're going to go into next. Talking about what we call feedback mechanisms and control systems. So the main regulatory process of homeostasis is called negative feedback, as you can see here in blue. So negative feedback is the main way that our body, and there are lots of these negative feedback pathways, there are hundreds and hundreds of them. There's as many negative feedback pathways as there are control conditions that are monitored within the body. And basically these control systems, these feedback systems, these negative feedback systems, they can sense a change in a control condition, a fluctuation in the internal environment, and they can bring about events that correct that fluctuation. So they send corrective feedback to different organs to help us bring our control conditions back to normal. And as long as we can fix that pretty quickly, our cells stay pretty happy. When we lose the ability to respond and to actually change things back to normal, our cells stop working properly. They are working at a subpar rate, right? And that we feel sick then. And that's what sickness is when our homeostatic conditions are disrupted. So all controlled conditions are at a set point. That set point has evolved in the different types of organism that we're talking about. So for example, in mammals, our body temperature is always 37 degrees Celsius or 98.6 degrees. No matter what's going on around you, you maintain that body temperature. Anything that changes your body temperature all of a sudden, if your body temperature rises to 40 degrees Celsius or drops down to 35 degrees Celsius, your body's going to respond through a negative feedback loop to send corrective feedback to bring your body temperature back to normal. So all control conditions in animals are going to be monitored and corrected using negative feedback mechanisms. They are common in metabolism, endocrine control, neuronal control, and muscle and circulatory control. So there are a lot of organ systems that are involved with this. The nervous system and the endocrine system are often the monitoring or regulating parts of the negative feedback loops. So let's look at what these look like. All right, so this is a image from an old textbook, and I like it because it's got kind of an analogy here. Here is what happens in the body, and here's the analogy that we can use. And we have negative feedback systems set up in all of our homes and buildings. This is what a thermostat is. So if we have a air conditioner, we're going to call it the effector, and we have a monitoring system, which we call the thermometer here in this instance, which is our receptor. The receptor's job is to basically monitor all day long, in this instance, temperature, right? What the temperature in the room is. If we have a set point to our controlled center, which is set at, let's say, 70 degrees, right? Then if the room temperature goes up above 70 or below 70, the receptor is going to sense that. And what the receptor then does is send information to a control center, right? The control center here is the thermostat, right? So the thermometer is often located in the thermostat, and its job is to monitor temperature. Temperature goes up or down from the set point, then the thermostat is activated, and it'll send a communication to the effector. The effector here is air conditioning, an electrical signal sent to the air conditioning from the thermostat, kicking the air conditioner on. It's called the effector. It's called the effector because it's going to bring about a response that brings us back to normal. It is the one doing the corrective feedback. So the air conditioner blows cold air, and that brings us back to normal temperature. Once we hit normal temperature, the receptor shuts off. It stops. It's not stimulated anymore. And if it's not stimulated anymore, then the thermostat's not stimulated anymore, and it turns off. And if the thermostat turns off, then the effector turns off, which is our air conditioner. We have a similar setup in our body. Notice we have parts to this, right? You can look over here in blue. We have a stimulus, or the stimulus can also be called a stress, right? And with body temperature, it's an increase or decrease with body temperature. We have a control condition, which is a body temperature, and that control condition is at a set point, right? So the control condition is set at a set point. The receptor monitors the control condition and the set point. If we go above or below the set point, it will send input to a control center saying, hey, temperature's going up or down. The control center will then send output to the proper effector, and then the effector will reverse or correct the stress, right? And bring our body temperature back to normal in the instance that we just used, right? So let's look in our body here. So the control center often in the body is the brain, right? Or the pituitary gland, or something that's part of the endocrine system. In our example here, the control system is the brain. We have receptors out in the body that are monitoring each of our control conditions. In this instance, it's body temperature. Body temperature is at a set point. The set point is 78. It's 78. It's 98.6 degrees, right? And let's say something happens to homeostasis. We get some stress that disrupts our body temperature. We start working out, right? And as we start working out, we start burning more glucose. Our body temperature goes up because metabolism has gone up. That increase in body temperature from an increase in metabolism, that stimulus is going to basically stimulate receptors. We have thermoreceptors, temperature receptors that are found in the internal core of our body. And they will respond when they pick up a change in our set point of 98.6 degrees. So if our body temperature goes up, it's going to send a nerve impulse, right? A nerve from the receptor. So you're going to have a nerve from the receptor run to the brain. So this is a nerve. Often this nerve is called a sensory nerve, right? So it's a sensory nerve. A sensory nerve is hooked to the receptor. It will be stimulated by the receptor, and an electrical current called an action potential will travel from the receptor into the brain. In the brain, this nerve meets another nerve or group of nerves that will then integrate that, saying, oh, body temperature has gone up. And then it will send the corrective output or proper output to the effector through another nerve here. So a second nerve comes out and goes to the effector, and it's called a motor neuron or a motor nerve, right? And this output, so we got input coming into our control center through the sensory nerve, and output going out the motor neuron from the brain to the effector. The effector here there are two different effectors in this particular instance. There are sweat glands and there are blood vessels in the skin. And in this instance, our body temperature has gone up, right? So it's gone up from 98.6 degrees, let's say, to 99. That stimulates the thermoreceptor. It stimulates a nerve that sends an action potential or electricity into the brain, into the thermoregulatory center of a part of the brain called the hypothalamus. And the hypothalamus takes that information and compares it to the set point and notices that body temperature has gone up. So it'll send the corrective outputs through a motor neuron to sweat glands, which increases the amount of sweat, and to blood vessels, which deliver hot blood to the skin. So you flush in the face and you start sweating, which will pull heat away from your core. And that will eventually bring your body temperature back to normal. Once your body temperature hits the normal, the stimulus stops stimulating the receptor shuts off, no more nerve action potentials going into the brain, no more output leaving the brain, and the system shuts down. That's negative feedback. That's how it works. Now, there is another type of feedback system that's called a positive feedback mechanism. So we know what negative feedback does. It is set at a set point, and it monitors that set point. And if there's a change above or below it, we reverse or send corrective feedback to bring us back to normal. So it reverses the disturbance back to normal. Here is your negative feedback, right? We don't have all of the pieces here, but you basically have a control center, right? A disturbance to the control center. Is sent in there through a receptor. The control condition is changed, right? The receptor senses that. We also have an effector here that is going to be stimulated to reverse the negative feedback, right? In between here, the control condition, right? Or our stress, our control condition, right? Our receptor senses this. We also have a control center, right? I'll just put CC for control center. That's between this. And I'll write over here, we have input, IN, and then output, right? From the control center. So you have input, IN, and output from the control center to the effector. The effector reverses the disturbance, right? Like we just talked about. Positive feedback does the opposite. Positive feedback increases the intensity of the disturbance, which sounds weird, right? Why would we want to do that? And obviously they're a little more rare in the body, but there are certain instances where a disturbance in our control condition actually is going to be increased, right? So that we get more of a disturbance. Some of the times physiologically where this mechanism is useful is with lactation, blood clotting, the immune response, and childbirth. So, for example, when the baby's pushing against the cervix in the female reproductive tract because the baby wants to be born, that stretches the cervix. There's a receptor in there called a stretch receptor, right? That is going to be stimulated, and it will send information to the brain saying, hey, the baby's pushing on the cervix. The output will be to the smooth muscles of the uterus to contract even harder, to push the baby harder. The more the baby pushes, the harder the contractions get. So you're intensifying the disturbance, right? Once you start blood clotting, you want it to intensify to block, right, the leaky blood vessel. When you get an immune response to COVID, you want your immune response to be large, right, to increase. So COVID causes the disturbance in homeostasis, and we increase the immune response to kill the virus. So there are times when a disturbance will actually change a control condition. The receptor senses it. It sends it to an effector through a control center that increases the disturbance or brings about positive feedback. So we increase the value of a set point. All right. Last but not least here are going to be regulators and conformers, right? So we're almost finished here with the first lecture. Conformers are invertebrates for the most part that lack backbones. And conformers always conform to the environmental conditions. So for example, if you look in the ocean, starfish, for example, and these annelid worms, their osmolarity of their body tissues is the same as salt water. If the saltwater osmolarity goes up, that is the amount of salt goes up in salt water, so do their body tissues. If the amount of salt goes down, so do their body tissues. They're very tolerant to changes in the environment because their body tissues have evolved to mimic the environment. That's what a conformer is. Now, the one thing that these animals do not have a very good job of doing is if salinity increases too much or salinity decreases too much, they typically are going to perish or die. They don't have any mechanisms to kind of fight that, or very few mechanisms. So they often live in very stable environments, like the ocean. A lot of these animals live in the ocean where the salinity, stays pretty close to the same all of the time, even though it increases and decreases in certain times of the year. And temperature might be more uniform. And so these conformers don't have to be exposed to extremes, right? Regulators, on the other hand, they like to be exposed to extremes. And the reason they can be exposed to extremes is because they have organs that perform biochemical and metabolic mechanisms that maintain their internal conditions using negative feedback. There are several invertebrate groups that are strict regulators, like vertebrates, like cephalopods. Octopi are very strict regulators. Some insects are very strict regulators, and so are decapods, these are crayfish. They are also very good at being regulators of their body fluids. That means their body fluid conditions stay exactly the same. They maintain homeostasis no matter what's going on out in the environment, whether there's an increase in salinity, an increase in temperature, decrease in temperature. There is a zone of stability or stableness in which they can maintain homeostasis very well. If we go above or below that zone, then these animals are going to have to conform or die. And most of these regulators have the ability to conform to a certain extent, and it's called acclimatization, which is on the next slide. So this shows you kind of conformers next to regulators. And so this is our variable we're talking about, our environmental variable in our internal environment. Is going to go up or down, and notice conformers will simply conform to that variable. They run the line of conformity. Here is what it looks like over here. The same disturbance in a regulator is going to stay flat. And this zone here where it stays flat is called the zone of stability, or the zone of stableness, is where I can maintain homeostasis. Notice when I get too extreme on either side, I have to conform. If I don't conform, then I'm going to die. So there are many regulators that are invertebrates. So decapods, like I said, crabs, lobsters, shrimp, and crayfish are really good regulators, as are mollusks and cephalopods and insects. All right. Obviously, vertebrates are great regulators, and conformers are most invertebrates. All right. So that finishes lecture 1. I will try to do lecture 2 or a part of lecture 2 tomorrow, and I hope you guys all have a great day.